Uh, Thank you, Dr. Miller, and thank you, Faizan, for taking us through what's happening at QCRC and also summarizing pluripotent cells starting from ES cells and to nuclear transfer in humans. You will remember that it was Ian Wilmot which introduced the concept of nuclear transfer, and you will remember Dolly the sheep, but now it's in humans, as you have heard from Faizan. He went on to tell us about IPS and the work of Yamanaka, but then he told us about indirect reprogramming. By that, he told us, thank you, Faizan, that you go down from a fibroblast, a somatic cell, to an IPS, and then differentiate it. First, de-differentiate, like he has shown you beautifully, and then differentiate, and that was the work of Yamanaka, as you have heard initially. And just to take the story further, I'm starting from the point where he stopped, and that is, again, what inspired the field was this work of Yamanaka, where he showed that you can de-differentiate a somatic cell using 4 transcription factors, and you see them, including c-MYC. The M stands for c-MYC, which is actually an oncogenic gene, which is really dangerous. And that has been modified, like you see here. Oh, I have to say, this lecture should have been given by Christophe, Raynaud, who had to travel, and you have to put up with me, so I apologize. I'm sorry. It's Christophe's work. So Yamanaka has introduced that, and it has never happened before that somebody gets the Nobel Prize within 8 years from the original paper, usually 20, 30 years, even in the case of the discovery of DNA by Watson and Crick. So it is unique. Yamanaka's work is unique because it's been validated. It's been taken further by that we're trying now to get rid of the retrovirus, which you have to use, or there are other viruses you can use. You use retrovirus, but there are other viruses which are less integrating, if you like. And even you can use small molecules and epigenetic modifications to produce IPS. My talk, however, will take the thing further, and I will introduce first what's happening in the field, and that's quite exciting. Rather than de-differentiate and then differentiate, which Yamanaka has started, you can differentiate to any type of cell, but in our case, we are interested in the cardiomyocyte. There is some work now showing, and that's the topic of my talk, or Christophe's talk, and that's direct reprogramming. Actually, Faizan has told you about that too. He used the same slide. And that you go from a fibroblast directly to an induced cardiomyocyte, direct reprogramming. How did that happen? I think that's the same slide, but how did that happen by, I will show you the person responsible and the transcription factors used, and I think the field owes a lot to Deepak Srivastava, and Deepak actually is a pediatric cardiologist, but he also works in the lab, and that typifies the cross over from the clinic to the lab. And Deepak has been offering to come with our group in the Chain of Hope to go to Africa because of his experience in pediatric cardiology. But what I'm talking about today is how he described for the first time, first in a mouse model, but later, as you would see, other people took it further to the human. And Deepak's first paper in Cell, and that was in 2010, not very long ago, described the GMT. What is the GMT? GATA4, MEF2C, TPX5. What are these things? They're transcription factors, and Faizan has told us that they are discovered from developmental biology, because these are the transcription factors in the embryo which actually determine cardiomyogenesis. So they are very specific to the heart, and he took advantage of that and used them to differentiate or make cells go straight from a fibroblast, which could be either a cardiac fibroblast or a fibroblast taken from the tail of the animal, or from other sources, from neonatal fibroblasts, foreskin in humans, for example. And then the work moved forward to show, in the original paper, Deepak showed convincingly that these cells produced by direct reprogramming without going into IPS to cardiomyocytes, and he gave them the name induced cardiomyocytes. It's not induced pluripotent cells anymore, it is direct skin to induced cardiomyocytes. And they actually integrated into the myocardium and started beating with the myocardium, both in vitro, but he went on to show also in another publication in Nature that you can do that in vivo, in the animal. And that's very exciting, because you know that after myocardial infarction, we get massive amount of fibroblasts. As a matter of fact, fibroblasts constitute nearly 50% of the cardiac cells before infarction, and a lot more after infarction. So you've got them there sitting, waiting to be reprogrammed, so to speak. It was Eric Olson, however, who took the field into the human arena, which is really quite exciting. And that was in 2013, only last year. What did Eric Olson do? He found that actually you need a lot more than GATA4, MEF2C, and TPX5, the original GMT, that HAN2, and interestingly, we knew for a long time that one transcription factor, MyoD, could move a fibroblast into a skeletal muscle. And so everybody said, oh, well, we're looking for a MyoD for the cardiomyocyte. Not so. But it was because Yamanaka introduced the topic of, we are going to use not one, like MyoD, but 4, maybe 5, maybe 6 transcription factors. That was Yamanaka's genius. So now, that's being applied, not MyoD, which takes you from a fibroblast to the skeletal muscle, that's easy. Or myogenin, which takes a skeletal muscle to what cell? Faizan, come on, tell me, what does myogenin do? Takes it to a smooth muscle cell. But in the heart, it's really much more complicated, and much more complicated in the human. So you need to add HAN2, for example, and that's a loop helix transcription factor. But then you heard yesterday from Despina that microRNA has been used for reprogramming. And that's what precisely what Eric Olson did. He used a lot of what Deepak has used, but added several other transcription factors and used microRNA, Despina's speciality, and succeeded in producing nearly 25% of the cells moving towards cardiomyocytes. I say towards cardiomyocytes because induced cardiomyocytes actually reproduce a lot of the characteristics of a cardiomyocyte, but not all of them. So they do have a lot of the molecules, like myogenin, troponin T, tropomyosin, and actually some beta as well, but they are immature, and they need, and you will hear from Gary, another member of the group later, how you can mature these cells. So this is the whole idea. Again, I already told you about, I'll have to speed up a bit because we are late, of how to induce the cardiomyocyte, fibroblast to jump into cardiomyocytes. What's happening at QCRC, and that's the work of Christophe. And that first is looking, where I was telling you about the endpoint for differentiation towards cardiomyocytes. Faizan told you how is that being done and using WINT. And WINT can actually differentiate the IPS up to 90% of them become cardiomyocytes. Wow! And they have strategies to do that. But here, with the direct reprogramming, Christophe and Faizan has been using several things. One, nanog. What is nanog? You've heard it from Faizan. Actually, it was discovered in Scotland as a stemness marker, and they called it nanog after a Scottish god. It's called nanog. But anyway, what our group has been doing is putting a variety of transcription factors, exposing the cells to these transcription factors. I said to you that the whole idea is to use more than one, but they've been using it at different times and for short periods, and that has been looking, at least the preliminary results, very promising in taking the fibroblasts into a cardiomyocytes. Now, the last slide, or the last two slides, is again Christophe and group, and our group here have been using STAT3. What is STAT3? STAT3, I will show you more, is a factor which actually induces more than one cardiogenic transcription factor. TBX5, GATA4, NKX 2.5, and this is just one pathway. What is that pathway? This is my almost final slide. The pathway is the JAK-STAT pathway. What is JAK? JAK is Janus kinase. Who's Janus? Janus was a Roman god who looked in both directions. And so Janus kinase is inside the cells and look outside the cell and inside the cell. What is STAT? STAT is signal transducer and activator of transduction. What does that all mean? Because using a ligand, you can activate STAT and Janus first, and STAT then forms a dimer, and the two together go inside the cell to actually not give you one, but three of the very important transcription factors which are cardiogenic. So STAT3 has been one of your toys in QCRC, after a publication obviously, but it is something very exciting. So I conclude by saying direct reprogramming of fibroblasts, which are in abundance both in the heart and everywhere else, offers extremely exciting opportunities for cardiac regeneration directly. And in vitro studies, as you have seen, constitute an important focus at QCRC. Thank you very much.